Welcome to this session, second session of the EMSCO meeting dedicated to collaborative biological studies. I'm MICHAELA Fontenay from Cochin Hospital in Paris. And I'm pleased to introduce three talks during the session. First of all, we are pleased to welcome Katharina GOETZE, who will present the biological studies appending to the LUSPLUS clinical trial. Thank you, Katharina, and the floor is yours.
Thank you, Michaela, for the very kind introduction. Salut, everybody. I hope you can hear me. I have a little bit of a cold, but I think it should work. So I'm very happy to give you a very brief overview of the collaborative studies that we're planning within the German, French, or European LUSPLUS trial. Just to give you a short reminder, luspatercept, which you're all very aware of, is the erythroid maturation agent, which works as a TGF-beta ligand trap. And based on the phase 3 MEDALIST trial data, which were published in the New England Journal almost 2 years ago now, led to the licensing of this and the approval of this drug in low-risk MDS with transfusion dependency and ring sideroblasts. Now, the LUSPLUS trial, of which I'm the coordinating investigator in Germany, and Lionel is the coordinating investigator in France. You can see here the protocol committee. So this is a joint effort from Germany, France, Austria, Switzerland, and hopefully also in the future, Spain will join. The design of the trial, just very briefly to let you know, it's a phase 3B open label single arm study because luspatercept is already approved. But the rationale for this trial is to exploit the maximum dose of 1.75 milligrams per kilogram to increase the response rate, because in the MEDALIST trial, there were about 40% of patients that really did not receive the maximum dose. We also plan to include a maximum of 10 patients which have failed HMA or lenalidomide treatment, in non-DAL5Q MDS. The primary endpoint is basically the same, red blood cell transfusion independency, according to the modified criteria at week 24, and we plan to include 70 patients from 18 centers. This trial, besides determining the response rates at the maximum dose, also has a large translational study program, which I'd like to present to you now. This is just very basically the trial overview. So the primary treatment phase is 24 weeks, and the response assessment is at cycle 9, and responders will be able to be treated within the trial for an entire year. Non-responders after 24 weeks or at week 25 will go off trial. So there are 5 work packages that we are planning within the translational studies. The first work package is immunophenotyping. So it's well known that in low-risk MDS, inflammatory mediators are upregulated, and we want to look at these inflammatory cytokines and also study the immune system compartments, so B cell, T cell, DC compartment, and also the MDSCs and the HSPCs and the macrophages. And this will be done by flow cytometry and on the Luminex platform using the secretome from these bone marrow samples from the patients and from serum samples. Work package 2, which is coordinated by Michaëla Fontenay, will look at the gene expression profile of SF3B1 mutated bone marrow mononuclear cells, and also on sorted erythroblasts, since it's known that there are splicing variants, or this is a splicing factor mutation. It's important to look at the gene expression and maybe be able to find a biomarker for response before and after treatment with luspatercept. It's well known already that there are niche effects of luspatercept and the sister drug sotatercept, which is not being continued. And it's been shown in 2013 that sotatercept reverses bone loss and disrupts the inhibition of erythropoiesis, which is mediated by the osteoblasts and osteoclasts. So this provides a rationale for studying the bone marrow microenvironment compartment in luspatercept treatment. And Uwe Platzbecker's group has recently shown that in the bone marrow, luspatercept also increases SDF1 levels in the mesenchymal stromal cells. So we have work package 3, which deals with the effects of luspatercept on the bone marrow microenvironment, also called the niche. And there are several aspects that we want to study in Munich and in Leipzig. And this first will be to do RNA sequencing on the sorted MSC populations from bone marrow mononuclear cells that are frozen from the patients before and after luspatercept treatment. We've optimized this protocol already in our lab, so we can actually do RNA sequencing on frozen MSCs and get decent numbers of reads with the 10x platform. The second point is to culture and expand these MSCs from the patient's bone marrow and do phenotyping analyses, look at their differentiation potential in terms of adipogenesis and osteogenesis, and a secretome analysis before and after treatment with the drug. And the third step would be to co-culture MSC before and after treatment with luspatercept from the patients with HSPC, so hematopoietic stem and progenitor cells, to determine the supportive capacity of these MSC in terms of HSC maintenance and also erythroid differentiation potential by flow cytometry and by very standard methylcellulose protocols. In work package 4, again, directed by MICHAELA, deals with the quantification of the ligand GDF11 and the ERFE variant that's produced by clonal erythroblasts, and this is a mass spectrometry project dealing with the specific GDF11 assay in the bone marrow and the plasma of these patients to see what the levels are and how they change during treatment with luspatercept. And the second part of this work package is the assessment of the ERFE variant production by these clonal erythroblasts and whether or not that changes during treatment with luspatercept. Finally, the work package 5, which will be conducted by Klaus Metzler in Leipzig, is mutational profiling of the bone marrow mononuclear cells at different time points before, during, and end of treatment with luspatercept, which has been well established in his lab, as you can see on the right side, for different mutations. And this is a targeted sequencing Illumina platform with the purpose of evaluating whether or not there is clonal evolution over time and whether there are additional mutations together with the SF3B1 and whether SF3B1 levels change during treatment. And I think with that, I'm actually already done, and I'll stop here, and I'm happy to take any questions.
Thank you very much, Katharina. Is there some question for Katharina in the audience? No, I have no question in the chat.
I'm Dr. Valcárcel from Spain. I have a question. Thank you very much, Katharina, for showing the study. Are you only looking for GDF11, or are you thinking in measuring also some other potential important ligands as GDF8 or some others that are supposed also to be involved in the activation of the TGF-beta pathway through SMAD2/3?
Thank you for this question, which is an important point, but I think I have to defer to MICHAELA for this because that's her work package.
Yes, at the moment we plan to specifically address a quantity of GDF11 because, but we will set up this technique using mass spec and immunoprecipitation of GDF11 with a specific antibody, indeed to discriminate between GDF11 and GDF8 that could be two candidates for the effect of the drug, to be targeted by this drug. So indeed, it could be interesting also to have the quantification of GDF11. And we keep, of course, the serum and plasma for that in case of negative result with GDF11, which could be expected regarding the last publication from Stefano. Thank you.
Thank you.
So for sure, if I can make one comment, this is an ambitious translational program. So we're very dependent on getting the bone marrow samples and the serum samples from these patients, which will be centrally banked in Leipzig and Paris. And we're very excited about being able to do these translational studies. So we hope you will also from Spain be able to join. I think there's a discussion with Guillermo, and it would be great to have you on board.
Katharina, can I ask you a question regarding the interaction with the microenvironment? Indeed, maybe we can probably have a new element, because your study are very powerful, I do think, because by looking at the MSCs, by the way you are, you will do it. So do you have any evidence for currently some modification of the MSC's transcriptome in MDS, in low-risk MDS at the moment, that could be modified by the drug, in your opinion?
So there's a lot of data from other labs out there, from transcriptome analysis from low-risk MDS samples, most of it from Mark Reimacher's lab in Rotterdam. We have just started doing the RNA-seq. We had some trouble with the frozen samples, being able to set it up to get enough reads. And if you take cultured MSC, you do get a real big change in the transcriptome due to the plastic adherence and the passaging of the cells. So we really thought it was important to be able to take frozen samples, because fresh is impossible, basically. But we're very confident now that we set up the protocol nicely that we can actually look at the transcriptome of the MSC from frozen samples with a decent number of reads. And we've done some pilot experiments on healthy, and it looks quite good. But of course, it's tricky, to be honest. And yeah, we'll have to see. But I think it's very worthwhile to look at the transcriptome signature of cells before and after the patients have been treated with luspatercept, especially if they're responding to the drug, because we're pretty sure that the inflammatory environment would be modified. So we expect to see some changes.
But in case of difficulties, do you think that it is acceptable to culture for a very short period of time?
Yeah, you can culture for passage 1 and then do the bulk sequencing from that. It's definitely a heterogeneous population. If you culture them, even if they're C271 selected, you're going to get a heterogeneous population. But if you standardize before and after treatment, then I think it would still be reasonable to get a good outcome.
Okay, thank you. So again, this protocol needs very good samples.
And we need to have spiculae. So if you get like the 3rd syringe from the aspirate of the bone marrow, you will not get any MSCs. Exactly. So that's a big important point that is kind of the bottleneck of this part of the project.
Yeah. Thank you. Thank you very much. So I would like to move now to the next talk, and I will present the protocols that Raphaël ITZYKSON set up for the IDEAL and IDIOME clinical studies in Saint-Louis Hospital, because he was not able to attend the meeting. So I will try to, yes, it is here, share my slide. Okay, so I think that you can see the slide. Sorry. Should go start again. Okay, so you heard this morning about the IDEAL and IDIOME clinical trials, so I will not come back to the design of these studies. This first slide shows you the banking, and in this protocol there is 3 cohorts. The first one is high-risk MDS with resistance to HMA. The second one is high-risk MDS who are naive of HMA, and the last cohort is made of lower risk MDS, and it is a smaller one. During the protocol, samples will be collected, bone marrow samples will be collected at inclusion after 3 cycles of evaluation and 6 cycles of evaluation, and after at progression. And the banking will be made in SOLARY Hospital under the coordination of Professor Emmanuel Clapier. Then in these protocols, there will be a number of collaborative studies that will be coordinated by Raphaël ITZYKSON. First of all, the sample will be indeed the sequencing of the gene will be of 60 genes will be performed at baseline and as a study exit. Using a classical targeted sequencing protocol that is currently used in the SOLARY Hospital by Professor Emmanuel Clapier. Then they will also address minimal residual diseases by digital PCR of the IDH clones. And then there will be several essay to address indeed both the transcriptome at single cell level when possible, combined with DNA sequencing. So for that, the team will use a target sequencing that is a single cell multi-omic techniques, and they will try to get both the mutational analysis in parallel with RNA sequencing from the same single cell, and this will be done on CD34 positive cells. And if this technique is not applicable, indeed they will use in a backup strategy with the Tapestry platform that is already used by Raphaël ITZYKSON and Mathieu Tischmann, and there is a paper in preparation. This Tapestry platform allowed to identify mutation or copy number variation at single cell level in cells which are identified by antibodies bound to the surface. And in this example, there is a combination of CD38 and HLA-DR, which help the recognition of the cells. And you can see here the identification of the mutation. These cells here are positive and those are negative. So this will compensate difficulties with the target sequencing that could be quite more tricky to use in this context of fresh human cells. And finally, they will also perform a study of the metabolism profile of the cells. And for that, they will use the CENIC techniques that has been set up by Raphaël Arguello with a scientist in CIML in Marseille. Raphaël is an immunologist, and he developed this technique using a short ex vivo phase in which the cells are incubated with various inhibitors of different metabolic pathways. And using these inhibitors and treatment with puromycins, it's possible to identify changes, metabolic changes in different cell populations that are identified by cell surface markers. And this technique has been described recently in a paper published in 2020. And indeed, this method is addressing the question of complex immunometabolic profiling. And it is possible to address glucose dependency, the fatty acid metabolism, the mitochondrial metabolism, and also the glycolytic pathways. So these techniques will be mixed together, and this work will be conducted by Giuseppe Di Feo, a young PhD students that joined the team of Raphaël ITZYKSON this year, and it is a European student that joined the team through the Intercept NDS ITN initiative. So that's it. I have nothing else to add, and if you have any question, I will try to answer, or if I can't, I will give the question to Raphaël, as that can probably answer better than I can do. Thank you. So I guess that there is no question. Okay. So I have a question from Guillermo. Does the CT allow us to include del5q patients? Is it for Katharina?
Sorry, MICHAELA. Yeah, the LUSPLUS trial is not for del5q patients. So it's only for non-del5q for the very rare situation where we have a non-del5q low-risk patient who maybe got treated with lenalidomide after AZA failure and then can go on the trial, but not for the classical del5q.
Thank you, Katharina. Okay, so no question for Raphaël. Okay, so maybe if Eric is there, we can move to the third talk.
Sure.
So Eric, can you share your screen? And Eric Solary will present some results of the DACOTA study that has been presented this morning by Raphaël, who was presented at distance. Thank you.
Yes, okay. Thank you and good afternoon, everybody. So this is the biological part of the DACOTA trials. So we had planned to collect samples from the blood and bone marrow during the course of this clinical trial. So this is what we got. At the beginning, we performed NGS screen of samples. All of them were not included because after a while, it was not always possible to include the patients, or the diagnosis was not confirmed. So we screened 231 patients, and 174 were included, of which 167 have NGS screen. So at inclusion, we received 167 samples of peripheral blood and 81 bone marrow samples. And then we receive additional samples after 3 cycles, 6 cycles at the end of treatment, and in some cases during the follow-up. So we can say that it works pretty well. These samples coming from Italy, Germany, and France, of course. So what we did, in addition to NGS screen, which was done every time we receive a sample, even when it took time to get from some hospital to our lab, Even in these cases, we were able to collect enough DNA to check the gene mutations. What was not possible in this case was to sort cells and keep them for further analysis. So this is the number of cases in which we were able to collect sorted peripheral blood monocytes, sorted peripheral blood CD3 positive cells, and in some cases also we collected the CD14 negative cells after having sorted these two other populations. We also kept the plasma from the blood and from the bone marrow samples, and when possible we sorted CD34 positive cells from the bone marrow. So this is the number of samples we collected at inclusion after 3 cycles, 6 cycles at the end of treatment, and during the follow-up. So these samples have in part already been used for some analysis, and others are stored in the lab. So what was done is first DNA sequencing using an NGS panel of 42 genes. These results have been sent to Raphaël. We also sent DNA and RNA from sorted monocytes to Ken Figueroa in Miami to perform methylation, DNA methylation analysis and RNA sequencing. So everything is done now. And we have more basic research performed on a few CD34 positive samples by the group of Françoise Porteux, looking at DNA damage, histone marks, and line mostly elements in CD34 positive cells again. So regarding the ERBS and the RNA sequencing, we did that in 3 parts, if I remember well. So we had some batch effect at some point, but we were able to correct the batch effect, and now we have all the information collected, and we need to connect these results to the outcome of patients, which we have not been able to do so far. We have been in touch with Yangzhou lab, who is a researcher located in Dallas, who is working on non-coding cis regulatory elements and enhancer dysregulation that could predict therapeutic response to HMAs. So far we sent samples we collected previously in the setting of other studies, one study that we published 5 years ago, and they are looking at these samples, but if you agree, we could share some DNA collected in the setting of the DACOTA trial to further explore the possibility that in non-coding regions there are some information that could be predictive of the response to HMAs. So that's of course open for discussion. We have also looked at inflammatory cytokine and chemokine. So some years ago we performed a first analysis together with Eric Padron in Moffitt with the Mayo Clinic, with Salt Lake City. We put together all the samples we had collected at that time, not in the setting of the clinical trials, but from everyday patients, I would say. And so we performed a first analysis showing a very heterogeneous pattern of cytokine level in the plasma of these patients, and validating an observation made by Klaus Gessler in Austria some years ago, actually in 1997, if I remember well. In a GXMed paper, he was already showing in CMML that a low plasma IL-10 level is predictive of a poor outcome in these patients for so far totally unknown reason. And it's totally independent also of the mutation pattern of any other parameter of the disease. So it's very surprising observations that we cannot explain so far. So we kept plasma during the DACOTA trials, so trying to further explore the cytokines. So this is the process we have used along the trial. We connected the supernatant and performed 2 centrifugations before storing the samples in aliquot. Then we use these samples to determine the level of cytokine and chemokine. So what did we see so far? again, we do not have the prognostic analysis of these observations. This is something we have to do now, but we have at least the observations that there are some cytokines that are overexpressed in CMML sample as compared to age-matched healthy donors that we always use to do this kind of studies. And so now we have to go into more details into the significance of these specific observations. We are also looking at the role of immature granulocytes in an independent study performed in our lab by Dorothée Selimo-Glubuet. So she's looking at the prognostic value of these immature granulocytes that behave as myeloid-derived suppressive cells. And by separating the samples of the DACOTA trials in poor and rich in these immature granulocytes, she was able to see correlation with abnormal levels of specific cytokine and especially in alarmins forming the heterodimer calprotectin. So you can see that in addition, the cytokine overexpressed in MDSC poor and MDSC rich patients are not exactly the same. So this is again something that we have to, whose meaning has to be deciphered. If we focus on two abnormally expressed proteins in the plasma of these patients, we observe that first, as I told you before, the calprotectin, which is the heterodimer between the alarmin S100A8 and alarmin S100A9, is overexpressed in DACOTA, in patients including in the DACOTA trials that demonstrated high levels of immature granulocytes. And in the case of CCL15, we can see here that these are many of the patients, independently of the level of the granulocytes, have very high level of CCL15, meaning that we have different type of deregulation of cytokine production during these diseases that we have to connect to other disease parameters. Some years ago, together with Valeria, with Omar Abdelwahab, and of course Ken Figueroa in Miami, we had been studying the predictive function of DNA methylation pattern in CMML. So we had identified molecular signatures that could be predictive of the response that we have to validate. We have a kind of epigenetic classifier that was proposed at that time. And we had also observed that by analyzing RNA sequencing data that some cytokine could be overexpressed in the patient's cells and correlating with the response to treatment. So this was based on RNA analysis in, again, sorted monocytes. So that was the observation at that time. So now, at that time also, on the left, it was shown that by adding recombinant cytokine to cell culture, we were able to inhibit the effect of HMAs with these specific cytokines. So, of course, now we had plasma, so we were able to look at these cytokines, and surprisingly enough, actually, in the blood of these patients with CMML, this cytokine is not overexpressed, but probably downregulated, which is a surprising observation, but again with heterogeneous levels, so we could probably further analyze what it means. Recently, we identified another inflammatory cytokine whose name is cytokine-like 1 or CTL1. We demonstrated that this cytokine was overproduced by leukemic monocytes, and through a kind of autocrine or paracrine loop was able to activate the MAP kinase pathway in monocytes and to further increase the production of CTL1. So there was a kind of positive feedback loop or autocrine loop that was also indicative of decreased ability of the cells to die by apoptosis, and we were able to target MCL1 and MAP kinase pathway to restore apoptosis and decrease the leukemic infiltration in NGS mice in PDX model of CMML. So during this study, we had identified that a fraction of CMML patients had a high level of plasma CTL1. So we looked at the level of CTL1 in the DACOTA cohort in 116 samples, because it needs a lot of plasma actually to do this dosage. And what we observed is that in the majority of cases, as in healthy donors, the level is 0, but in about 30% of the patients, there is an increased level of the CTL1 level in the plasma, with 3 outliers with very, very high level of this cytokine. And we have now to understand what it means in terms of disease outcome and response to treatment. Finally, the last one we have looked so far is named IGFBP2, insulin growth factor binding protein 2, which was shown to be overproduced by the bone marrow niche in the context of MDS. So in CMML, we detected an overexpression of this IGFBP2 in a global cohort of 259 CMML. So you can see it again, it's very heterogeneous, but it's much higher in CMML patients, either in MDS or MPN CMML patients. So we looked at that specifically in the DACOTA cohort, and we obtained actually very similar results to those obtained in what we call the myeloma cohort in our lab, which is every CMML samples we can collect at diagnosis, and you can see that the proliferative nature or the severe nature of CMML included in DACOTA does not modify significantly the IGFBP2 plasma level in these patients. So what is ongoing with this specific cytokine is that we are trying to identify the cells that are overproducing this cytokine, the impact of this cytokine on hematopoietic differentiation, and the clinical significance of this overexpression observed in some patients. So to summarize, what we have done with the samples collected from the DACOTA trial is that we have been able in almost every patient to explore 42 genes by using our NGS myeloid panel. We perform RNA sequencing and methylation analysis on sorted monocyte DNA, and these results are now there, and we have performed the measurement of plasma level of the indicated inflammatory chemokine and cytokines. So we need now to connect with the statisticians, which is not an easy task, but we are waiting for a green line from the Saint Louis statistician to explore the significance of all these biological information. We need to get access to the database to correlate this data to the clinical and biological information we have get. And we have exploratory projects that I mentioned during this talk with Françoise Porteux, Dorothée Selimoglu-Buet, and Ryan Xu. So that's it. I would like to acknowledge, of course, all the people in the lab that have been doing this work along the years, but also all of you, the physicians that send us the samples, and the organizers of these interesting trials. Thank you for your attention.
Thank you, Eric. Is there any questions for Eric?
I have some,
Yes.
if I may. Impressive work, Eric, as usual. I have a question. So the data you showed are all baseline, isn't it?
Yes, exactly. So far, we have used only the samples at baseline.
Yeah, and you said that, yeah, you said that you compare with age-matched controls for a.
Yes, that's what we do when we measure, for example, cytokines. So actually, for almost every parameter, we try to have age-matched healthy control when we can.
And then I have another question that is puzzling me for a while. So we are dealing here in the DACOTA with high-risk CMML, blast or proliferative. And my question is, what about inflammasome in these patients? Because it should be a little bit different from the one we see in, let's say, dysplastic lower risk CMML. And again, decitabine is known to stimulate some inflammation anyway, short term or long term. So how can we see what is done by the decitabine treatment, or what is baseline, and what is really important in the different settings?
Well, so it's of course a very good point. What we have done so far is to analyze the samples before any treatment. So we cannot suggest that the treatment is involved in the observations we made. That being said, the pattern of inflammatory chemokine and cytokine is highly heterogeneous, as you have seen, and this is still poorly explained and understood. So that's the kind of work we have to pursue prospectively, I think, in the future trials, to try to get more understanding of the meaning of that. So far, we don't know whether the different patterns of cytokine secretion we have observed have a significant meaning in terms of outcome of the patients under treatment. This is something we will check, of course. And we could now look at the samples we collected after 3 or 6 cycles to see whether there is an impact of the treatment, as you mentioned, on the level of cytokine. So that's something we could do now. Yes, sure.
And you don't have the clinical outcome yet?
No, not yet, not yet.
Over.
Hello, Eric. I have a very simple question, and this is transportation time and feasibility of sending you samples from all across Europe. What about quality? Were you satisfied with it? Any ideas for improvement? Because I mean, we talk about all very sophisticated and very intriguing data, but of course also the pre-analytic quality of the samples is very important. So is that something where you would make some recommendations for future clinical trials if we centralize translational research? I think this is important.
So that's, of course, a very, very important point. So regarding the cells, I think that a 24-hour delay, and even in some cases 2 days, but 24 hours is good. We can collect the cells, and the quality of the cells we collect is good. We have not too much cell death. So for the cell collection, the 24-hour delay is appropriate, and we can work this way. And we can centralize, I think, the cell samples. Regarding the plasma or serum is probably less obvious. We probably lose information by collecting the plasma more than 24 hours after cell sampling, after blood sampling or bone marrow sampling. And the best thing to do, I think, in the future, but I guess MICHAELA is going to speak about that in the afternoon, but it would be to collect on site the plasma or serum. And probably on site and the day of the sampling, because probably we will have more reliable information at the end.
Okay, Eric, can I ask you a question? I was quite surprised by the result published regarding IL-10 and the low level of IL-10. Was it in the peripheral blood or in the bone marrow in this paper?
It was in the peripheral blood. Both times, it was shown by Klaus Gessler. It was confirmed in the paper we did with Eric Padron some years ago. And so in the DACOTA trial, I don't know so far, of course, but even more, I think the Austrian people at the beginning of or at the end of the last century, a long time ago, they tried to treat patients with IL-10 actually, and they treated 4 or 5 patients with IL-10. So we have very limited observations, but nevertheless, it was the idea that it could interact with GM-CSF hypersensitivity of progenitors, and well, it has not been confirmed and never extended. So we cannot make any additional comment on that.
Yes, my question was related to the production of calprotectin, because some are thinking that I-10 could be an inducer of calprotectin production. So you have very high level of calprotectin, which could be expected regarding the default of maturation of your cells, but should be related to something, and probably it's not to to I-10.
I am not sure we looked at the correlation between these two specific cytokines, but we can check that. That's a good point. Sure.
Okay, thank you. So we have 1 minute. We have to stop there because it's time for the next session. And I would like to thank the participants and you for listening. Thank you.